A main focus of research in my laboratory is on potassium channels, using potassium channels as targets for treating autoimmune diseases. And we've really looked at two different avenues. One is focusing on a type of white blood cells, T lymphocytes, that play a role in a number of autoimmune diseases, including multiple sclerosis and rheumatoid arthritis. What we have found is that on T lymphocytes that play a role in autoimmune diseases, we have a high expression of a potassium channel called Kv1.3. This channel is present in a much lower number in other T lymphocytes in those patients. We have identified, in collaboration with really a worldwide team, a C anemone peptide that blocks this potassium channel Kv1.3. We have modified it to make it highly potent and highly selective for this potassium channel. And what this peptide does is that it plugs the Kv1.3 potassium channel on T lymphocytes. So it inhibits the function of the T lymphocytes that play a role in autoimmune diseases. But it does not affect T lymphocytes that target, for example, influenza virus or other acute infections. In animal models, this blocker can treat models of multiple sclerosis and rheumatoid arthritis, but does not prevent the clearance of acute infections from viruses or bacteria. This peptide has been licensed to a company called Kineta in Seattle, and they have conducted phase 1A and phase 1B clinical trials. And the results have been very exciting so far. The peptide is well tolerated in those healthy volunteers, and this peptide is being pursued. But in addition to this avenue, we're also looking at another potassium channel as a potential target, specifically for rheumatoid arthritis. So while the first project focused on the immune system in T lymphocytes, the second project focuses on synoviocytes found in joints. And during rheumatoid arthritis, those synoviocytes are activated, and they're extremely destructive. We have identified in recent years a potassium channel called KCA1.1, or the BK channel, on those cells. And we have been able to block this channel, to block a lot of the very destructive features of those cells. And more recently, we have been able to stop the progression of 2 animal models of rheumatoid arthritis using this blocker. And in the long run, we're really hoping to develop this as a non-immunosuppressive therapy for rheumatoid arthritis.